these are D, which is B and C, right? So this is a carboxyl group and an amino group, hence amino acids. So remember, all amino acids. Okay, for those who are wanting to talk about other stuff, you're welcome to, but please take it outside of the class, because it's really hard to talk to everybody. Thank you. Okay, so all amino acids, remember, have your central carbon. Attached to hydrogen on each end, we have the amino group and a carboxyl group, and then we have the R groups, which is what varies. Okay. Oops, sorry about all of that. Okay, if the cytoplasm of a cell is at pH 7 and the mitochondrial matrix is at pH 8, then the concentration of hydrogen ions. Okay, let's first think about this. Is hydrogen ion, if you have a higher concentration, is it more acidic or basic? Acidic, right? Is your pH lower or higher? Lower. Lower, right? So if you're going, the concentration of hydrogen ions is going to be higher or lower in the cell or the mitochondria?
Higher in the cell.
Higher in the cell, right? So let's see. And it's going to be how much higher? 10 times, right? So sometimes higher than what? So it's D, right? 10 times higher. I think everyone a lot of people got confused on this one. Again, that 7 is actually more acidic than 8, because this was commonly missed, but I think a lot of you probably understand, right? Hopefully that pH is, it's on a gradient of a log scale, right? So it's 10, you're increasing concentration by 10 every pH you move up or down. Okay, so the R group or side chain of the amino acid serine is as follows. The R group on side chain of the amino acid leucine is as follows. Where would you expect to find these amino acids in a globular protein in aqueous solution? Okay, so first, let's look at these R groups. What do you notice? Are they both polar, non-polar, or is one polar and the other non-polar? Right. So which one is polar? So oxygen, having oxygen on there makes it polar. So what makes something polar, you guys? Right, so having unshared, unequally shared electrons, right? So when you have a covalent bond, remember what two atoms are really highly electronegative? Nitrogen and oxygen, right? So when oxygen forms a bond with carbon or hydrogen, it's going to hog those electrons, making it polar. It's going to have that partial negative charge. So here on serine, you have this OH group on the end, which is one of the functional groups, right? That also states that it's polar. So this is a polar R group, and here we have all carbons and hydrogens. So kind of a hydrocarbon, right? Is a hydrocarbon polar or non-polar? Non-polar, right? Because carbon and hydrogen share their electrons equally. Okay, so if you're on a globular protein in a cell, where would the polar R group tend to face? In towards the center of the protein or out towards the aqueous solution of the cell? Out, right? Because water is polar. So these R groups that are polar tend to move towards the polar aqueous solution of the cell. And a non-polar R group most likely will be basically pushed towards the interior of that protein, as it doesn't have any attraction to water. So I think it's A. Leucine would be on the interior and serine would be on the exterior. Do we need to clear that up anymore? Does that make sense now? Yes?
So that was the right answer, or is that speculation?
A is correct. Yes. Which of the following is not true? Both chloroplasts and mitochondria. Right. They are not part of the endomembrane system. So we look at chloroplasts and mitochondria. They both have the double membranes on the outside, around it, right? So outer and inner membranes. They both have their own DNA. They both have their own ribosomes. But remember the theory is they came from their endosymbiotic theory, right? So they were engulfed by these cells a long time ago. We don't really know where their membranes come from, whereas all of the other endomembrane system, where do they get their membrane from? Yes, the endoplasmic reticulum. So the nuclear envelope, the endoplasmic reticulum, the Golgi apparatus, lysosomes, the plasma membrane, those are all part of the endomembrane system. Chloroplasts and mitochondria get their, we don't know their origins of their membranes, other than it's believed to be they were engulfed, right, a long time ago and became symbionts. Okay, so which structure is not part of the endomembrane system? So both these were missed. Which one's this going to be? Chloroplasts, right? So again, they have to be either physically in contact or basically all this membrane is made here in the ER. So the smooth ER synthesizes what for the most part? Lipids, great, and the ER synthesizes, the rough ER synthesizes a lot of proteins, right? So all the membrane comes from this lipid synthesis right here, and it pinches off and it joins the Golgi, it can join the plasma membrane, it can form lysosomes, things like this. Okay, a salamander relies on hydrogen bonding to stick to various surfaces. Therefore, a salamander would have the greatest difficulty clinging to a hydrocarbon. Okay, so hydrogen bonding, right, occurs between two polar molecules. So again, oxygen and hydrogen don't share their electrons equally. So there's a partial negative charge on the oxygen, a partial negative charge on the hydrogen. Same thing happens in ammonia, right? Nitrogen's hogging these electrons, so it has this partial charge, and this slight charge, right, like it'd be kind of like weak magnets attract each other, so it bonds together. So that's what this salamander, is relying on, right? So is a damp surface, is that polar? Yeah. Yes, water's polar, right? So it wouldn't have a problem there. Mostly carbon, nitrogen. Okay, is nitrogen electronegative? Yeah. Yes, right? So that's a polar molecule there. So it shouldn't have any problem clinging to that. And then carbon-oxygen bonds. Are those polar? Mm-hmm. Yes, no problem there. But hydrocarbons again are what? Carbon and hydrogen bonds. Those are non-polar, right? So that would be a surface that the salamander would not be able to cling to because it can't form those hydrogen bonds. Questions on that one? Here's a picture of some examples of hydrocarbons, right? Okay, what is the difference between an aldose sugar and a ketose sugar? A. Well, and B. Number of carbons. Yeah. No, sorry. I was thinking of a different question. Sorry. The position of the carbonyl group, right? So it can either be on the very end, right? And it'll be aldose, or it can be second to the end where it's a ketose. So I know I said in class at least once, make sure you know what defines a ketose from an aldose and how these sugars are defined, right? So the number of carbons is what I was going after, and I thought that question was asking. Number of carbons and where this carbonyl group is placed. Okay, a hydrocarbon skeleton is covalently bonded to an amino group at one end and a carboxyl group at the other end. When placed in water, this molecule would function. C, right? It's going to be an acid or base. This is exactly what our amino acids are. So this was, again, being familiar with this functional chart, right? So the carboxyl group can release this hydrogen. When you release a hydrogen solution, you're acid or base? Acid, right? So it can act as an acid if it has this on it. An amino group can take on an extra hydrogen, right? So if you're pulling a hydrogen out of solution, are you acid or base? Base. A base. So if you're a molecule that has this on either end, like an amino acid, you can act as an acid or a base. Which molecule shown above contains a functional group that cells use to transfer energy between organic molecules? Organic molecules. So the answer is D. Phosphate, right? ATP, energy, right? I know that's corny, but my friend teaches high school science. That's what she does. Okay, so ATP has these 3 phosphate groups on it. Remember we talked about this? It's in your chart, your functional group chart. So this is a really electronegative functional group. It's got all these oxygens on it, and it can react with water. When it does this, it can release energy for the cell to use, right? So our cells pretty much use ATP for energy in almost everything, either directly or indirectly. Okay, which of the following is not a polymer? Yes, glucose. I was really sad, you guys, that this was one of the more commonly missed questions because we went over polymers and monomers a lot, I thought, right? What is a polymer? Right, it's a large molecule made up of repetitive monomers, which are either the same molecule or something very similar, right? So polysaccharide is a polymer, a carbohydrate polymer that's made up of monosaccharides, which glucose is a monosaccharide, right? And we have nucleic acids, proteins are also polymers. So DNA, what does that fall under?
Polymer.
Right, and it's a nucleic acid. So I think the most common answer to this question other than A was D. So some folks got confused and were thinking that DNA was not a polymer. But it is, right? It's made up of those nucleotides. What do nucleotides have? What are the 3 components?
Pentose sugar, nitrogenous base.
Right. That pentose sugar with a phosphate group and a nitrogenous base. So they're repetitive nucleotides bound together. Okay, one of the primary functions of RNA molecules is to function in the synthesis of proteins. So DNA is the nucleic acid that's responsible for, it's basically the blueprint to all of our genes, right? So it passes on hereditary information, it can synthesize itself, right? But RNA is made from DNA, and we're going to go into this a lot deeper later this semester. But RNA is basically used in our cells to help with the synthesis of proteins. So, DNA is read, and it's transcribed into RNA, and then this is read by the ribosomes, right? Ribosomes are made up of RNA, and they make proteins. They bind those amino acids together. Motor proteins provide the molecular motion in cells by interacting with what type of cellular structures? A, cytoskeleton, right? So talk about these motor proteins. They can bind to different things in the cell, so vesicles or even organelles, right? And use ATP, and will move along some part of the cytoskeleton. Here's a microtubule. We talked about how microtubules are used along with these motor proteins for flagella, cilia, and how microfilaments are used to move they're responsible for the amoeba's movement, cytoplasmic streaming, and all this uses those motor proteins, right? Which level of protein structure do the alpha helix and beta pleated sheets represent?
B. Secondary.
This was secondary, right? So what type of bonding is occurring in this form here? Hydrogen bonds, great. Between what part of the amino acids? Is it the backbone or the side chains? Backbone, right? Okay, so this is secondary structure, the hydrogen bonds between this backbone, which is the amino acid carboxyl group in carbon, right? Either form the alpha helix or the beta pleated. So the tertiary comes from different types of interactions, bonding between those R groups, right? All of the following are part of a prokaryotic cell except? Correct. No. See? No. What's the main difference between prokaryotic and eukaryotic cells?
Plasma membrane.
Sorry?
The plasma membrane?
No, both of them have plasma membranes.
Ribosome.
Don't look at the answers, just
I
what's the
don't-
main difference
we don't-
between a prokaryotic cell and eukaryotic
This
cell?
one stumped me.
Right, so they have membrane-bound organelles, so nucleus, eukaryotic cells do, right? Nucleus, mitochondria, endoplasmic reticulum. Prokaryotic cells don't have any membrane except for their outer membrane. Remember, they have their DNA that's not really, it's not membrane-bound. It's just kind of joins in an area called the nucleoid, but there's no membrane around it. So that's the main difference. Endoplasmic reticulum is a membrane-bound organelle. We find that in eukaryotic cells, not prokaryotes. Prokaryotes do have a cell wall.
So no internal membranes?
Correct. Yep. Okay, so which of the following statements is false? Saturated fats. Well, I have to read the answer, sorry. Okay, it's not A. It's not B. No, it's D, right? So D is not, so saturated fats have no double bonds. Okay, and this means that C is true because whenever there's a double bond, carbon can only make 4 bonds, right? So the carbon is releasing one of those hydrogens to bind with another carbon, it's going to have less hydrogen. So let's look at it here, right? So saturated fats, we find these commonly in animal fat, and they have, right, they both have triglyceride, and then they have 3 fatty acids coming off here. So in saturated fats, there's no double bonds. That's why they're called saturated. They're saturated with hydrogen. Every carbon is going to be bound to another carbon on each side, but then every other place is going to have a hydrogen filling in. So all of these white circles here represent hydrogens. And remember, at room temperature, they're typically solid because they can compact real tightly. Whereas unsaturated fats, they have these double bonds present at least in one of these fatty acids, which causes a kink, right? And that makes it not be able to pack in as tightly, so it's typically liquid fluid at room temperature. And where this double bond occurs, right, if we have the same number of carbons, so if I have a fatty acid that's 5 carbons long, and there's going to be hydrogens off of every one of these, right? Carbon bonds with 4 on each side and so forth. And then we have one over here, 1, 2, 3, 4, but there's a double bond here, right? And there's hydrogens. So 1, 2, 3 can only have a hydrogen here. This is going to be, no. Right? So 1, 2, 3, 4, 5, 6, 7, 8 hydrogens for this one. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 hydrogens for that one. Does that make sense? One of the buffers that contributes to pH stability in human blood is carbonic acid. Carbonic acid is a weak acid. When placed in an aqueous solution, dissociates into the bicarbonate ion and a hydrogen ion as noted below. Okay, so remember buffer systems help maintain the pH in cells or in a solution, right? So they can go back and forth. So this can either dissociate to add hydrogen here if it needs to increase acidity to keep it balanced, or this can bind to the hydrogen and pull hydrogen out of the solution to help it balance the pH that way if it needs to be. Okay, so the pH of blood drops, becoming more acidic. So that means what's happening to the hydrogen ion? Is it decreasing or increasing? Increasing, right? One would expect what to happen in this equation? A. Correct, A. Right? So if there's more hydrogen being added into the cell, so this is our cell. I'm not going to draw anything about hydrogen ions. So let's say that typically there's 2 hydrogen ions in this cell, and all of a sudden something raises the acidity or lowers the pH, right, we add some hydrogen in here. This buffer system is gonna pull these two out to maintain a pH of 2. Hydrogen here, right? So this ion is gonna bind to these to pull them out, which means this will increase and this decreases the concentration, but it's A. Does that make sense? Okay, great. Oh, okay. And that was the majority. So I put every question on here that 50% or more people missed. Like I said, if you work and you cannot make office hours during the day, you're welcome to come up and look at your exam right now or next week, but email me if you're gonna do it, we need to do it next week. If you can come on my office hours, that's preferable, so we don't have to bring people tonight. Okay, so you guys have questions?